Hello, my name is Holger Mamersopova and I'm here to present an introductory lecture on antibiotic resistance, the grand challenge of today. First, I would like to start with some history. In 1928, Alexander Fleming discovered a substance released by yeast in contaminated petri dish which kills surrounding bacteria, and he named it penicillin. However, penicillin became broadly available only in 1945 due to Dorothy Hodgkin, who investigated the chemical structure of penicillin by X-ray crystallography. Penicillin V, an oral form, was developed in 1952 and a synthetic form produced in 1957. This laid foundation of a new era of synthesizing antibiotics. Let's have a look at the structure of bacterial cell wall. Bacteria can be divided into gram-positive and gram-negative stems based on the cell wall structure. The cell wall consists of a lipid bilayer membrane and a mesh-like peptidoglycan sheath or matrix. Gram-positive bacteria builds a thick peptidoglycan matrix around a single lipid membrane, whereas gram-negative bacteria builds a thinner peptidoglycan sheath between two layers of lipid membranes. In this presentation, I will focus on gram-positive bacteria. Water constantly enters bacteria by osmosis and puts pressure on the lipid membrane. Peptidoglycan sheath or matrix provides a structural support to the membrane and prevents bacteria from bursting. Penicillin prevents the bacteria from building such a peptidoglycan matrix. Now let's discuss mechanisms of action of penicillin. Here in this picture you can see cell membrane and peptidoglycan structure of the bacteria, and this is the enlarged form of it. Peptidoglycan is made of small building blocks of two sugars connected with amino acid and a short peptide bridge extending to the side. These sugars form chains which are then cross-linked to form a peptidoglycan matrix. An enzyme called penicillin binding protein is responsible for assembling these chains into matrix by creating crosslinks. Penicillin blocks this enzyme by directly binding to amino acid serine in the enzyme's active site. The part of penicillin which does the action of blocking is called beta-lactam ring. Thus, beta-lactam ring opens and forms a bond with serine within the enzyme and blocks from its action. Examples of antibiotics with similar mode of actions are amoxicillin, methicillin, and ampicillin. Let's look at mechanisms of antibiotic resistance. Many antibiotics are effective in treating many infections. However, bacteria are living organisms and as such adapt to survive and protect themselves from antibiotics and develop resistant mechanisms via genetic modifications. For example, MRSA, a strain of Staphylococcus aureus, evolved to express a different enzyme called penicillin binding protein 2A with an altered active site which does not interact with beta-lactam ring, making beta-lactam antibiotics ineffective. Bacteria can also express a special enzyme called beta-lactamase, which specifically binds the beta-lactam ring and destroys it, making beta-lactam antibiotics ineffective again. A similar example of expressing NDM1 gene, which codes for New Delhi metallo-beta-lactamase 1, which can break all known beta-lactam rings. In addition, this kind of resistant genes located on small plasmids of bacteria can be passed to other bacteria both horizontally or vertically through generations. And finally, let's discuss important points in fighting the antibiotic resistance. Let's focus first on misuse and overuse of antibiotics in humans, which led to antibiotic resistance to begin with. The second would be inappropriate use of antibiotics in agriculture. Hens, for example, hens are fed antibiotics to increase their body mass. In fighting the antibiotic resistance, it is important to consider developing new classes of antibiotics effective against bacterial resistance, or packaging antibiotics in inhibitors which block bacterial degraders. The last but not least is strengthening of our immune system with healthy lifestyles and eating habits. Thank you for listening, and now we can proceed to a small quiz on Kahoot.